great pleasure. It's an enormous pleasure to have Ed Rubel here. Ed is one of the most important scientists working in the field of development of the auditory system. He did his PhD before Neuroscience time, so it was called Zoology and Psychology. And then eventually made his way to the Washington University in Seattle, where he is now. And he made a huge number of great discoveries. You will see some of them in his lecture. And one of the most important one, is the generation of hair cells. Unfortunately, not in humans. Or not yet in humans. So, Ed is really he's one of the greatest scientists working today in the auditory system, and I'm really happy that he's here. I think the first time we were in Israel was 2 years ago, a year and a half ago in the Easter meeting. All of us who were there and heard him talk, I think, remember it until today. And then he so enjoyed his visit that he decided to come for another 2 months. So he's now at the beginning of his visit, and those of you who want to maybe to talk and interact with him in the next couple of months, he's here in Haifa. Okay, Ed.
Thank you. Well, thank you. It's really a great pleasure to come back. I found Israel so fascinating, and found out so much of myself by coming here, about myself by coming here. I decided that I had to come back I was here for 1 week, and I had to come back for 2 months, so maybe next time I'll come back for 2 years, okay? I will talk today about, I'm going to do the worst thing possible.
Can you put the microphone on?
Oh, yeah, I can put the microphone on. I was actually told to put the microphone on, and I already forgot. Is it on now? Yeah. Okay. So I'm going to do the worst thing possible. I'm going to give a new lecture, okay? And nobody should ever give a new lecture. Please feel free to stop me at any time during this lecture. If you have questions, I can come back and answer them and so forth. So I want to talk about the regulation of dendritic patterning by synaptic activity in binaural brainstem neurons. First, I have to do the obligatory, here's Seattle, and we always have to show a picture of our home too. And this is a Space Needle here, Mount Rainier, which is a wonderful mountain to climb, and it's a beautiful place surrounded by water on both sides and mountains on both sides. So those of you that haven't visited it should. It's one of the prettiest cities in the world. Okay, the topics I'm going to talk about, I'm going to talk a little bit about binaural hearing. I'll then introduce you to Nucleus laminaris in all its grandeur. How many people in the room have ever heard of Nucleus laminaris? Well, there's at least one. Okay, a few. Okay, I'll talk about the specificity and signals underlying the regulation of dendritic size and structure. I'll then talk about some static and dynamic studies revealing sites of regulation, that is, where is the regulation happening, and the influence of pattern. And then I will talk about potential cellular mechanisms, if you will, and its relation to human health. And since I can't define the cellular mechanisms, that will be mostly sort of guesses, and I call them teasers. And at some point in time, you or I or somebody else will put them all together, and we will understand cellular pathways whereby dendrites are regulated and how this might relate to human health. Now, here's the problem with binaural hearing. Okay, it's a really interesting problem. This guy, you can see him. He's listening to this guy talk, and he's wondering why all these women are talking to this guy rather than him. Okay, this guy's probably doing the same thing over there, but we can't see him. And the woman might be saying, well, why am I talking to him rather than this guy? The point is that this is a noisy crowd, a noisy area, and this person can focus in on one particular voice and sentence stream in this noise. And they have to be able to do this quite accurately and pick out the spectral properties as well as the noise, and as well as the temporal properties of that thing. So we know a little bit about how that happens, and we know a little bit about why it happens. What is binaural hearing for? Well, its purpose is our sound localization. In some species, like a nocturnal animal, depending on which way the wind is blowing, this is the only way they can find predators or prey, or know where the animal is. More importantly for humans, it is sound segregation. That is, knowing where a sound is coming from. When we enter a room, what are the voices, what are the acoustical properties of the room, and so forth, and computing that in our head. And finally, it's for, and most importantly for language, it's pulling signal out of noise. And we get, and remember decibels, how we measure sound intensity, are a logarithmic scale, and we get about 15 to 20 dB of improvement in terms of signal-noise ratio out of the fact that we can hear with two ears and we can integrate that information. The primary cues, of course, are interaural time differences, which we call ITDs, which work primarily for low-frequency sound. This is an oversimplification, obviously. And interaural level differences, which work primarily for the high frequencies in the spectrum of sound. And the way this works, of course, is here's a listener. I would have put a chicken in here, but I didn't have one. And when a sound is coming from straight ahead, obviously there's no difference in terms of the time of arrival at the two ears, and no difference in the intensity at the two ears. But when the sound is from the side, there's, in a human, up to about a 0.6 milliseconds or 600 microsecond difference in time of arrival of the sound in the two ears, and a difference in the high frequencies at about, say, 10 kilohertz of about 20 decibels in terms of sound intensity. So our brain can compute these different properties and tell where the spectrum is coming from, or a particular part of the spectrum is coming from. And note that say in a smaller animal like a bird, or with a small head, that goes down to 50 microseconds, or in fact about 100 microseconds. Birds have a specialized mechanism between their ears to make their head bigger, functionally bigger, and so it's really, even though if you do the actual difference in terms of distance, it will only be about 30 or 50 microseconds, but they have this specialized mechanism to stretch that out to 100 microseconds or a little more, which we can talk about later. In the rest of the talk, I will only talk about interaural time differences and how this is computed, and the cells that are primarily involved in computing it. This is probably the most important cue for language, of course, because most of language is in the low frequency range. So here is a schematic of a gerbil, a mammal, a typical mammal brainstem. The human would have all this stuff down here in terms of the pyramidal system, the cortical pyramidal system, but we have the eighth nerve coming in, synapsing in different parts of the cochlear nucleus, and then this binaural excitatory pathway where introventral cochlear nucleus cells project to the single cells project to the ipsilateral MSO and the contralateral MSO, but to different dendrites. And so these MSO cells are getting excitatory input from both ears, okay, matching excitatory input from the same frequency region or tonotopic region of both ears. And they're also getting inhibitory input through the medial nucleus of the trapezoid body. Now we look at the chick brain, and here's the brainstem. Cerebellum would be up here. Fourth ventricle is here. Eighth nerve does the same thing. It goes in, synapses on a group of cells known as magnocellularis, nucleus magnocellularis, which is homologous in every way that's ever been found to the anteroventral cochlear nucleus for the devotees, the bushy cells there, and the nucleus angularis, which is in some ways like the dorsal cochlear nucleus and posteroventral cochlear nucleus. Its output forms these big calyx-form endings, excitatory endings, the eighth nerve it does, and single cells in nucleus magnocellularis then send a bifurcating axon, one branch going in a parallel set of fashion to the ipsilateral nucleus laminaris, to the dorsal dendrites, and the other set going across the midline and forming this unique delay line, if you will, and going to the ventral dendrites on the opposite side. So the cells in nucleus laminaris are getting excitatory input from both ears to the different dendrites. They're also getting some inhibitory input from the superior olivary nucleus going to all three of those nuclei, if you will. Now let's look at the cells in MSO and nucleus laminaris. Can we turn down the lights a little bit? That's perfect. Is that okay with everybody? Now here in the chick, we'll talk mostly about the chick. Here are a bunch of cells stained with an antibody against MAP2, which nicely stains the dendrites, as you can see, as well as the cell bodies. And you can see this unique lamina of cells that would actually be going over there and there, and the dorsal dendritic area and the ventral dendritic area you can easily see. Similar kind of structure in nucleus in the medial superior olive with the cell bodies, not as beautifully lined up. They don't need to be in a gerbil for some reason. But here we see the dorsal dendrites, or actually these are the lateral dendrites, and the medial dendrites, and you can see they're pretty symmetrical in both cases, okay? If we look across a number of different species, a number of different terrestrial vertebrates with elongated cochleas, here's alligator, okay, and we see the dorsal dendrites of nucleus laminaris and the ventral dendrites of nucleus laminaris. Here's a chick, a little bit bigger segment that I showed you, and I'll go more into this gradient of dendritic length in a moment. Okay, here's a gerbil. I showed you some cells in the gerbil before. Here's the gerbil, and here's the human. Okay, this isn't a MAP2 stain, but it shows pretty much the dendrites of human. And you can see across the vertebrates, there's this similar structure. It's a unique structure with segregated dendrites that are getting different inputs, and the job of the cell is now to compute when those inputs are coming in at the exact same time, and for different cells in different parts of the nucleus, there's going to be a difference in that window of coincidence detection based on interaural time differences that are computed by those cells. So they will be activated best when that time window is achieved, and you have phase-locked inputs coming into both dendrites of the same cell. And we can go into in questions about how the biophysics of that works, but that's not what I'm going to talk about now. What we have is a system where we can do some unusually clever experiments, or unusually well-controlled experiments. So let's look at the chick. Here's a chicken. If we look at its brain, it looks something like this. Here's the cerebellum, brainstem, forebrain, optic tectum, and so forth. Take off the cerebellum, and we take a slice through there, and what we get is something that looks like this, in just looking at it in optical field. And here's nucleus magnocellularis. Here are the eighth nerve fibers coming in. Here's nucleus magnocellularis here. The output of nucleus magnocellularis going across the midline to the ventral part of nucleus laminaris, this clear space, and ipsilaterally dorsal part. If we use a few antibodies on it, we get something like this. Here's nucleus here's the midline again, nucleus magnocellularis. These fibers coming out, doing this loop that I'll talk about here, coming into the dorsal side, shown here. Here's the cell body lamina, and the other ones from the other side coming out and synapsing on the ventral dendrites of nucleus laminaris. Okay, so it's a unique structure. I showed you the organization, but let me stress it. Eighth nerve comes in, bifurcates, form this big caliform synapse, excitatory synapse, okay, that are getting phase-locked action potentials, which means it's responding with interspike intervals that match or are at some subharmonic of the tonal properties that it's responding to, and sending two axons out, one that forms this delay line such that different axons, different cells get input at different delays, and the other axon going out and forming this parallel set of terminals. What this means is that we can manipulate the input to just one set of dendrites, say just the ventral dendrites, or we can manipulate the input to the dorsal dendrites on one side and the ventral dendrites on the other side. And since all of this apparatus is monaural, we can leave the other inputs responding in a normal way and thereby study the actual synaptic surface that the input is coming to, independent of the synaptic surface of the other one. So if we lay Nucleus laminaris down and look at it from the top, we know that the Tonotopic Organization is something like this, with these cells in the posterior lateral part receiving low-frequency information, and progressively more rostromedial cells receiving high-frequency information, and this delay line goes across, is isotopic, and it goes across an individual isofrequency line where the spread of terminal fields in this dimension along an isofrequency line is all the way across the nucleus, and the spread in this dimension is only about 2 cells wide, about 20 microns wide. So it's a set of parallel inputs you can think of that we can manipulate. If you look at Nucleus laminaris again, you see this unique frequency gradient, or this unique dendritic gradient, such that the cells way over here at the high frequency end of the nucleus are responding to very, very high frequencies, and they have little short dendrites, as I'll show you, and these respond to lower frequencies, and they have big long dendrites, and that dendritic organization exactly follows this tonotopic organization. And if you ask me why they have that, I can go into a bunch of theoretical reasons, but nobody really knows, and but it's obviously important. Here are better pictures of the cells. These are from individual cell fillings. These are photomicrographs. And here's the low-frequency cell, a mid-frequency cell, a high-frequency cell, and three-dimensional reconstructions of their dendrites look like that. And so it's a huge difference in dendritic surface area. That's an order of magnitude in size following that frequency gradient. We could look at that in individual cells, and I want to point out one other property of those cells. Here's a high-frequency cell and a low-frequency cell. I'm going to make the high-frequency cell bigger. Notice these have lots of little dendrites coming out. These have only a few. But also notice when you're in the plane outside the isofrequency plane, you got that flat dendritic sheet, sort of like a Purkinje cell in the cerebellum, and conforming to this isofrequency organization. So what do we want to do with those cells? Well, what we want to do is understand how synaptic activity regulates dendrites. That's the point of our studies. First, does it, and does it in unique ways? What's the time course in yeah? No, the surface area, your question's about the surface area of the dendrites, right? Yeah, the surface area of the dendrites follows the total dendritic length, and I'll give some properties of that. But the bottom line is that we published this a long time ago by doing surface area measurements. These areas have about these cells have about an order of magnitude greater surface area than the high frequency cells. Okay, it's also shorter dendrites, more individual primary dendrites, but about an order of magnitude difference in surface area. I think of that as synaptic contact area, obviously. So what we want to do is understand how dendrites are regulated and how specific is that regulation when we change activity. One of the things I didn't tell you, and I should have, is that these are the only excitatory inputs that these cells get. There's a unique system that has to do microsecond timing, so it doesn't like to have a lot of other inputs because that would screw up the timing. So these are the only. If you take out both ears, these cells become totally quiet. If you leave the ears in place, there's a huge amount of spontaneous activity that's all due to the Brownian motion of the stereocilia of the hair cells. You can quiet the hair cells in a variety of ways, and all the spontaneous activity goes away here, and if you do it to both ears, all the spontaneous goes away there. So if you're thinking cortex, this ain't anything like cortex, okay, because we're controlling all of the excitatory activity to one membrane surface. It's still getting inhibitory activity. So if we look over, if we cut the dendrites, cut the axons to the ventral dendrites, and then look at various times afterward, so what we've done is deafferented these dendrites, the ventral ones, and left the input normal to the dorsal dendrites. Okay, what we see here's a control cell. These were old studies done by the Golgi method, and here's 16 days post-track cut. We call this a track cut. Okay, and you can see this cell has become highly asymmetrical. There is a little bit of change in this time. They actually grow spines. These cells are spineless normally, but they actually grow some spines on the dorsal dendrite. But that's the only change we've seen in the dorsal dendrite. If we look at the time course of that change, note that these are hours, okay, and these are days. So we're looking at the percent change of the deafferented dendrites from cutting midline, a millimeter away from the dendrites along that axon, and we're seeing like a 20% change in the absolute size of that dendrite within 2 hours. So very, very rapid changes in the size of that dendrite. And that was done in a number of ways. It was done not only by measuring them in Golgi preparations, but done by electron microscopy, looking at the volume density of dendritic surface area. And here's the time course of the change. Now this is a percent difference, or the percent change in dendrite per hour in the first hour, the second hour, the third hour, 2 to 4, and so forth. And you can see there are big changes and then very small changes. These big changes are fast retraction of dendrite, decreased microtubule density, looking at from electron microscopy, a thin dendroplasm that's obvious when you look at the organelles in it, and this dendritic surface, membrane surface, seems to sort of float back into the cell body, so you actually get an increase in soma size, okay? That increase in soma size goes away, it stabilizes, and you get continual decrease, but a slow level of decrease in dendritic size. So from there, what we decided to do was exclusively use single-cell electroporation so that we could study the size of each dendrites and study them at various times after we made a manipulation, either statically or dynamically. So we basically use electroporation. We can take a slice. These are from about a 5 to 10 day old chicken post-hatch. And by the way, chickens start hearing at 11 days incubation. They are hearing virtually adult-like by the time they hatch, around 21 days of incubation. And by 5 days after incubation, their sensitivity is adult. 5 days after hatching, their sensitivity is adult-like. So we can take a slice, we can electropate any number of cells. Okay, in this one we electroporated 4 cells. We only look at the ones that have their complete dendrite dendritic surface within our slice, and then we study those either dynamically or statically at various times after a manipulation. Whoop, I like this picture. So here you can see our cell filling goes all the way out to the tips of the dendrites and so forth like that. So let's look at what happens quantitatively at various times after we do these various manipulations. If the cell we're going to change symmetrically after we cut the ventral input, here we'd expect the whole cell, all the dendrites to get smaller, and I've already told you that that doesn't happen, that just the ventral dendrite gets smaller, right? And so we'll just look at this in the first 7 hours after a manipulation. And we can compare that with what happens when we don't actually damage that axon, but we take out the cochlea by stopping activity, and we know we've stopped activity because we've measured there, okay, and look at it 8 hours after cochlear removal. What we would expect, of course, hypothesize, is that on this side of the brain we'd get a shrinkage of the ventral dendrite, not of the dorsal dendrite, and on this side of the brain we'd get the opposite. We get a shrinkage of the dorsal dendrite. We can do a third manipulation where we can put TTX in the ear, thereby blocking the activity of the auditory nerve, and we'd expect the same kind of change if it's activity-driven. But to make sure we're not doing any damage, we can let the TTX wear off, see if auditory thresholds come back to normal, and then look later and see if that change corrects itself. And finally, we can mimic that whole situation by taking a slice preparation and electrically stimulate one auditory nerve or both auditory nerves and look at what happens to the dendrites on both sides of the brain, both the dorsal and ventral dendrites. So those are the experiments I'm going to tell you about next. Okay, here's an example. Okay, in a normal cell, we get what we call a dendritic change index, okay, and that's equal to 4, where we're just looking at the relative size of the two dendrites, okay, how much change there is. And here, after a track cut in 80 hours, we're getting an index of 23, okay, and we see that there's a shrinkage of the ventral and no change in the dorsal dendrites, as we said before, and we get a change of about 30% in 8 hours of the dendritic size. If we do another manipulation, like cochlear removal, we get approximately the same, maybe a little bit less change, but these are obviously not statistically different. In the deprived ventral dendrite, that would be in the opposite side, and a similar change in the deprived dorsal dendrite, no change in the undeprived dendrites. Similarly, if we put TTX in, we get a similar size change in 8 hours, and we don't on both sides of the brain, and no change in the dorsal dendrites. But we can also let that TTX wear off, and by 24 hours we get normal hearing, we'll look at it 48 hours later, and we see there's no significant difference between the two sides of the same cells. That is, this is not significantly different from 0, nor is this, obviously. So the dendrites have recorrected themselves, and this is a really important kinds of phenomena, or important result, because anytime you want to do a manipulation like this, and you're getting a deprivation effect, you really have to try and understand, are you just creating a pathology, or is this some sort of plasticity that would naturally be possible to occur? If that wasn't correcting itself, that is, if that dendrite didn't regrow when activity is restored, then we think we're just creating a pathology. The fact that it can regrow and recorrect itself. And the answer of, did we study the actual size of the dorsal dendrites? Did we do bilateral manipulations? The answer is yes. We studied the actual size of the cells, and this is a summary, and it's in the paper, and we did bilateral manipulations. Yeah?
Sorry, I might have missed it. Would the presynaptic terminal dissociate from the shrinking dendrites?
No.
Following the shrinkage?
They stay on. They stay on, and they don't change in size that we can see. We have some work that we did, not in Nucleus laminaris, but in Nucleus magnocellularis that we did a long time ago, showing that the presynaptic terminals may get snipped off by glia expansion. We see at the same times in magnocellularis, when we do this or this, we see a decrease in the number of terminals on the cell and an influx of glial filaments in there. And we think that that's happening in Nucleus laminaris, but we don't have any clear proof of it. Okay, we've measured terminal size in a EM paper that we published several years ago. We actually measured terminal size, and the terminals don't change their size, so we must be getting less terminals there. Okay, so we measured the coverage area of the membrane that's covered by both the excitatory terminals and the inhibitory terminals. We can tell their difference. The inhibitory terminals, we don't get any sprouting of inhibitory terminals. Their density gets greater compared to the excitatory ones, obviously, but there's no sprouting. Pardon? We haven't tried that. Presumably we could. It'd be very difficult to do that in vivo in a meaningful way, but we can do that in an in vitro preparation, and actually have done some of that in vitro preparation. It's not in these slides, but it's in a couple papers published with Stacy Sorensen in, I think, 2007 and 2011. So we have studied this dynamically. And so here's a Nucleus laminaris cell, okay, that we've electroporated, and we can then watch individual dendrites grow and shrink over time. And you can, I think, see that one growing and shrink. There is some z-axis movement here, but we can correct in that when we measure. And when you do that, what you see, in this case, that dendrite has been deafferented. And what you see in a normal animal is, if you look at the difference in total branch length from the manipulated dendrite versus the unmanipulated dendrite in a control slice, you don't see any difference in change, but or any change in that difference. But in a deafferented slice, by 1 hour, there's almost a significant change. By 2 hours, there's a highly significant change in the relative size of, in this case, the ventral dendrite of a cell. And we typically see that big a change. This is one cell, but we typically, this is a whole group of cells with that manipulation.
Why is the last error bar larger on the right? Because there are fluctuations?
Yeah, I'm going to show you that in a minute. Okay, that is, these cells are always growing and retracting, as you can see. And what we did is we measured, and I don't know why that one in particular is maybe because it's longer out there and they're responding more differently, okay? But I don't know. If you looked at that, and, well, tests of variance, as you probably know, are not very robust. And so it's probably not statistically reliable, although it might be. I mean, it's 2 to 3 times. But that's a good question. So this is going back to some static imaging, both from Stacy's studies, and I guess I have the slides in the wrong order. Both from Stacy's studies and these static imaging studies, we wondered what part of the dendrite is changing. Is it the shaft? Is the whole dendrite pulling in? Or are we changing it in terms of individual segments, okay? And so what we can do is we can trace each individual segment of the cells in our three-dimensional reconstructions, and or we call them individual branches. At every branch point, we start a new branch, and we can measure the amount of change that's going on in the whole dendrite versus just the secondary and tertiary branches of the dendrite. And what you notice here, okay, this is in a bunch of preparations in which we did that, and we look at, in this case, a ventral deprived dendrite or the dorsal deprived dendrite, we see approximately the same response in the just measuring the tertiary branches as we do in the whole, if we measure the whole dendrite, which suggests that in fact this response is occurring in these individual branches, the secondary and tertiary branches, not really in the main shaft, that this is a movement in and out of the cells. And this fits with what Stacy found in her dynamic imaging, in which what we did was we looked at various dendrites in control animals, in dorsal dendrites, control ventral dendrites, intact dorsal dendrites, and deafferented ventral dendrites, and we looked at the number of dendrites, or the percent of dendrites that have remained stable in each 10-minute epoch versus the number that grow or shrink. And when most of the dendrites remain stable, okay, a large percentage remain stable, but what we see when we do a manipulation is in the number, in this case, that retract, okay, a percent of retract grew, and the percent that grow went down. I think that answers the question you asked there. So what we're getting here is in these secondary and tertiary branches, we're getting everything's growing and retracting, and what we're doing is changing the balance. And that's actually shown here in doing shoal, what are called shoal analysis, where you build concentric circles around your cell body, and you measure the percent of dendrites that are in each concentric circle. And you can see that shifts. The percent of dendrites that are only a few microns away from the cell gets greater, and you lose the ones that are longer or further from the cell body, which is what you'd expect in that respect. So there's one other question that interested us, and that is, well, basically, is this a function of the pattern of stimulation or the total amount of stimulation that a cell is getting? So we have a unique situation here where we can change both the pattern of stimulation to the cell or the total amount of stimulation that we're giving. And the way we do this is take a slice preparation, which we know pretty well mimics in terms of time course and in terms of the amount of change to what happens in vivo, and we can stimulate, we can give no stimulation. When we give no stimulation, these cells in terms of spikes, okay, and I'm not talking about miniatures, I'm talking about spikes, these cells are quiet, okay. We can also put stimulating electrodes, and in this case we stimulate at a relatively low rate for these neurons at 10 hertz over a few hours, okay, and the cells respond to every stimulation, and we stimulate both auditory nerves in this case, and we find basically no significant change in the dendrites, okay, in either case. So with no stimulation, we're not getting a significant change, and with bilateral stimulation, we're not getting a significant change. And here we're looking at individual dendrites, of course. Okay, and now we do the same preparation, and we stimulate one dendrite, and we look at what happens. Okay, and on this side of the brain, on the ipsilateral side of the brain, we see that the ventral dendrite, in this case, we're stimulating this side of the brain. So now what we're doing is stimulating the dorsal dendrites and not stimulating the ventral dendrites on this side of the brain, and they shrink. Okay, this is not a significant change. If we look at the other side of the same brain, we see the opposite effect. Okay, The dorsal dendrites shrink and the ventral dendrites don't. We have done the studies that you asked earlier of whether we can prevent this or can we modulate this by putting glutamate agonists or antagonists on, and we can to some extent, but the cells don't like them after a while, which is understandable because these cells have unique firing properties such that they don't just depolarize continuously. If you put glutamate on, they'll give one spike, just like if you inject them with a DC current, they'll give one spike and then they'll shut down because you activate as soon as you depolarize these cells, you activate these huge outward currents that basically shunt the cell. Okay, so they have this unique physiological property. So the conclusions to this part of the talk are that Nucleus laminaris has structural and functional specializations that are highly conserved in low-frequency hearing terrestrial vertebrates. They all look the same, from alligators to man. It's a useful model in which to investigate dendritic regulation by afferent innervation and synaptic activity. Synaptic regulation of dendritic structure is highly localized and dynamic and depends on the balance of growth and retraction of tertiary processes. And rapid changes in dendritic structure are fully reversible, i.e. it's not a pathology, and appear to depend on the balance of activity of the different membrane surfaces, at least at short times. I wouldn't count on that for long time intervals, personally, as opposed to total amount of input. So I want to end by talking about what I call teasers, what may be happening in these dendrites, and what's its importance. Okay, we've shown, and Larry Trussell has shown, that calcium is independently regulated in the dorsal and ventral dendrites. If you stimulate the dorsal dendrites, you get a calcium response there. You get some calcium response in the ventral dendrites, okay, but not nearly as much. Okay, this has been done with Fura. It has not been done with modern calcium sensors, but both Larry did a nice study on that, and we did a study on that as well. Interestingly, and not surprisingly from that, when we did our electron microscopy studies well before that, and we looked at the changes in the dendrites, what we saw in the deafferented dendrites was a clear quantitative change in the number of microtubules and in the volume density of microtubules and the volume density of neurofilaments by 2 hours after we started the differential stimulation. Also, and I'll show you a little bit about this, these dendrites have an extraordinary high number and distribution of various calcium modulating proteins, particularly plasma membrane calcium ATPase 2, PMCA2, which is involved in extruding calcium from the cell. Okay, so it seems that calcium is being highly regulated in these dendrites, and that's important. And when you do a deprivation or deafferentation, you get an almost immediate change in PMCA protein, which is interesting. So also a variety of calcium binding proteins and ion channel proteins are independently regulated in the two dendrites. And the Fragile X protein, Fragile X metal retardation proteins, or FMR1, is found highly expressed in these dendrites, which makes sense. Here's PMCA2, for example. Okay, here's laminaris. This happens to be the high frequency end, so you get little short dendrites, and you can see that it's very, very highly expressed in the dendritic region. If you look and you double label with MAP2, you see it's just covering the MAP2 region, indicating it's in the dendrites. I should say, however, that you find PMCA2 both in the axons and the dendrites. If you do the EM autoradiography, you find more in the dendrites. Interestingly enough, okay, this happens to be a track cut, but you can see the same thing if you do a cochlear removal. Here's PMCA2. This would be the cell body lamina here. And here's PMCA2 symmetrically in the dendritic lamina. If you do a track cut and look a half an hour later, and there's no degeneration by that time, axonal degeneration, what you see, and these are blood vessels, by the way, what you see is a clear difference in PMCA2 on the two sides of the cell. By 3 hours, it's a dramatic difference. So if that protein is what we're seeing is the amount of membrane protein of PMCA2, which we don't know, that could be a reason why we see an increase in calcium. We could be seeing an increase in calcium. In any case, it's not regulating calcium nearly as well in terms of that part of the cell. We also see, of course, a variety of potassium channel proteins. This happens to be Kv3.1, a high voltage activated potassium channel. Here you see the two sides of the cell. It's highly regulated. Okay, in the dendrites, here's a high power showing its membrane properties, membrane surface properties. And here we did a cochlear removal, and you can see within 3 hours there's a huge decrease on the ventral side and a considerable decrease. By the way, this recovers. This recovers. It's a temporary decrease in the amount of protein, and it might not be a decrease in the amount of protein that's membrane bound, or the amount of the activation of these channels. Interestingly enough, here's here's Fragile X protein. This is MAP2 staining, and here's the cell body lamina, and here's Fragile X protein. Now, I'll remind you that Fragile X protein is an RNA binding protein, and thereby is regulating translation, and thus it can regulate translation very, very rapidly and very selectively in individual parts of the cell. It's not very many. Now, we know Fragile X protein is in a lot of dendrites. Fragile X disease is considered a type of autism, and it's considered by many a dendritic disease, having people that looked at cortex. And Fragile X mental retardation and other kinds of autism are usually thought about as cortical diseases, that is, as cognitive diseases, so they must involve places like cortex and hippocampus. But we see huge amounts, in fact, higher amounts of Fragile X protein in Nucleus laminaris dendrites than in most cortical dendrites, interestingly enough. And if we look carefully at where the Fragile X protein is, okay, where it is in dendritic bifurcations, interestingly enough. Here you see MAP2 and a little pocket of Fragile X protein in this dendritic bifurcation. Here's another one, here's another one, here's another one. And that's a common thing. We recently looked at some mammal brains. This is very recent, unpublished data. And we see it also, here's the cell body lamina. I'm sorry, this isn't as well stained. And here you see Fragile X protein. And here you see it in the bifurcations of the MSO dendrites. So what we have here is a system where many, many proteins are changing. Hence, it's not surprising that the dendrites are changing in a very selective way. And we have all the machinery for changing these proteins. I didn't mention that, of course, cytoskeletal proteins are some of the major proteins that are regulated, where translation is regulated by Fragile X, and ion channel proteins are regulated in dendrites and known to bind to Fragile X protein. Their RNA is known to bind to Fragile X protein, as is the RNA for cytoskeletal proteins for many types. Now, I should say that there's in the binding studies, there are approximately 2,000 proteins that can bind to Fragile X RNA. I'm sorry, 2,000 RNAs that can bind to Fragile X protein. So which ones are actually, it's actually controlling translation in any particular neuron type is still up in the air in our system as well as others. Now, the other thing we might ask, is this important for communication, or is this relevant to human communication? Well, we know that in Fragile X disease, Fragile X mental retardation, and in other types of autism, most people have normal thresholds but have huge communication problems, particularly in noisy environments. This has not been, in most cases, this is in Fragile X, I can't find a single paper actually looking at binaural processing in these patients, but it's known that they have problems, big problems with communication, and that those problems are particularly important in noisy environments. We also know that human MSO shares the cellular structures and connections and proteins with the chick nucleus laminaris, and I'll show you a little bit about that. And finally, the human MSO appears to be abnormal in people with autism spectrum disorders. We don't know about Fragile X disease, and I'll show you a little piece of information on each of those, which leads us to at least hypothesize that, in fact, the communication problems of people with autism have to do with the information getting to the cortex, or much of it has to, some of it, I should say, has to do with the right information getting to the cortex as opposed to cortical processing in itself. And I'll show you a little information on that. Here's a test that was done in a paper that was published in 2005, which is one of the few that I've found that actually do anything in terms of testing binaural processing in autistic adults. Okay, what these people did was they looked at the ability in noise of people to localize speakers that were at the end of a distribution and to pay attention to just those speakers. They looked at reaction time and some other behavioral measures, okay? And some of those data are shown here. Okay, this is the percent of responses that were correct to the different, to the end speakers in a circular array or semicircular array. And what we can see is the autistic group, the responses were pretty much, or kind of similar to all the speakers, okay? But in the control group, the responses were much greater, significantly greater to the target speakers than the other speakers, okay? And this is in a noisy environment, okay? And this is the reaction time, and reaction time basically shows the same thing. That is that there were low reaction. The Fragile X people, the autistic listeners were pretty flat in terms of their reaction time, whereas the control group, when they responded to a speaker, that's sort of in the middle or toward the middle, they took a long time as if they weren't sure, but when it was out on the edges, the normal people took a shorter time than the other people. Interestingly enough, they also measured the N1 potential of these people and the control group and the autistic group, and they found a much bigger N1 in response to the target speakers than the other speakers, whereas the autistic group didn't show this response. Let's get to a little neurobiology here. This is a human brain. Human brains, remember, are very good at localizing low-frequency information, and they have a very, very nice MSO. If you try and look for an MSO in a mouse, you can't find it because they're a high-frequency animal. And in a rat, you can hardly find it. But here's MSO in a human, a nice line of cells like I showed you before. If you look at high power, here it is. Here's the MSO in a human. Okay, and here's what the cells look like. This data was provided by Randy Kaleza. We then collaborated with Randy and looked at a variety of proteins. This shows you KV3.1, the one I showed you in the chick brain, and you can see it's very, very highly expressed in the cell bodies as well as the dendrites of the human MSO, showing that they're basically doing the same job, using the same proteins as the chicken and the gerbil. And here is Fragile X protein in the dendrites. Now we can't get the resolution that we did in the gerbil and the chick because the brains aren't fixed for quite a while and so forth. But these dendrites of the human MSO are loaded with Fragile X more than most other cells, any other cells in the brainstem that we've seen. So again, having all the same things. Now the last slide I'll show you is from Randy's collection, and it's his only brains he's gotten of people with autism. He doesn't have any with people with Fragile X disease. But here's the normal human MSO, and here's MSO in 3 different autistic people. And as you can see, this nucleus is totally misshapen. In some people, you can't even find it. Okay, in others, it looks terrible in terms of the cell bodies. In some that it's just distorted and like that. Now, it'll be very interesting to get some people with Fragile X disease as well. So, what we can say here is that, in summary, is that if you look at this cell group across vertebrates, from reptiles to birds to man, it seems to have all of the same characteristics, which isn't very surprising because these cells have to do the same thing. They have to encode tiny differences in time of arrival of spikes, and so they need to use a certain set of proteins, and they're highly specialized for doing that. It also looks like these cells, in terms of their dendritic structure, are very, very highly regulated, and that this regulation can happen quite rapidly. Whether it happens quite rapidly during normal processing and during normal life, we don't know. It also looks like that this nucleus, if it's not functioning well, will lead to severe problems in communication, and we're starting to understand some of the reasons why that would be true. So there's much more work to do, but I think it shows, one, the conservation across vertebrates and how you can use the brain of a lowly chicken to maybe understand diseases of communication in man. So I want to acknowledge that this was all done in Seattle. This is still a beautiful place. This is a view from my porch, which you're welcome to visit when you're in Seattle. We'll probably be there. The 2 people that did most of the experiments are Stacy Sorensen, who is now at the Allen Brain Institute in Seattle, and Yuan Wang, who might be looking for a job very soon, and she's just like the best postdoc I've ever worked with. Len Kazmarek and Meili Brown were the people that got us interested in Fragile X protein and Fragile X regulation of other proteins. Randy Kaleza is a great collaborator who I've actually never met, but has provided us with brains, and we provided him with antibodies. And of course, this has all been supported for the last 40-some years or 30-some years by the National Institutes of Health. So I'd like to thank you. For your attention and open the floor to any questions. Yeah.
So let's say you suppose that with some delay between the 2 sides, there will be no plasticity, but if you will cause bigger and bigger physiological delays between the two sides, you will see plasticity?
You know, that's a really good question, because these cells are very, very active cells, and I don't think that you would see plasticity in these cells if you just change the. I don't think there would be enough change in activity if you just changed the interaural time in a non-physiological way. I think if you actually cut down the activity, say you have a hearing loss, a mild hearing loss, and so you're cutting down the activities. Say you're losing your endocochlear potential, so you're losing spontaneous activity in one ear, or something like that. I expect that you would see these kinds of changes. But in terms of just changing interaural time, I don't think you'd get enough of a change in activity. We have no evidence to date. We have no evidence against this, but we have no evidence to date that throwing the interaural times off of off the right timing will cause a change. The other thing we don't know, remember, is if these changes are due to a change in, when most people think of activity, they don't discriminate between spikes and membrane activity. There are many people that think that regulation of dendrites has more to do with miniatures than spikes and the ongoing membrane changes, and we haven't tested that here adequately. Okay. Yeah.
So, activity has a relatively short time to suggest that maybe this process serves some purpose in healthy animals, maybe some form of adaptation. Do you have any thoughts on that or some speculation?
Of course, I have speculations on everything. I don't know. Okay, I think that this system is what we really call a very private system in the brain, and it's very, very useful for studying how dendrites are regulated. Okay, but I don't really have a feeling that in ongoing behavior that this kind of plasticity is doing anything. I think it's a model of what can happen in other systems where you do get a change in activity on specific membrane surfaces. So I was at a Keystone meeting last spring, and I presented some of this material, and Roger Chen beat me up pretty severely because he said, well, what does this mean for the animal's behavior? And I said, I don't have any idea, and I don't really care. Roger didn't like that answer at all because he wanted me to do what people do in cortex. And what I'm trying to do here is trying to find out some basic parameters about how dendrites are regulated, not trying to predict the behavior. This Fragile X information came as a big surprise to me, obviously, but that's a pretty severe kind of thing, and it relates very, very well to the ability of the people to use binaural cues. How that might be important for the variability that we see in the normal population, I'm not at all sure. Yeah. Yeah.
I think this is a mechanism, and I want to ask whether you did any experiments in stabilizing biological with drugs like paclitaxel. That's one. Two, did you try to block protein synthesis and degradation? Because there is enough time for that to occur.
Right. Right.
And the third question is, look at membrane turnover.
Did we look at membrane turnover? Yeah. And the answer to all those is no. I think those are great experiments to do. They really depended on us. So we've worked on these dendrites very intermittently over the last 20 years or 30 years when I could find somebody that had enough patience to draw individual dendrites and things like that. It had to wait until we could effectively electroporate individual cells. We tried to do a lot of that kind of experiment with sort of bulk imaging with MAP2 and things like that, but there's enough variability within the cells and with your cutting angle and stuff like that that it really had to wait until we had good ways of reconstructing individual cells and electroporating individual cells. We only just recently developed, it's published, ways of doing genetic manipulation and expressing things like GCAMP in chicks, and we now do have a way of doing that. That was just published this past December, I think.
What we've done is to put the primary capsule, you will see that the robust changes in the length of the dendrites will not occur.
Well, I think that's a great experiment, but
You don't have to have genetics or
right. I think you're right, but I really wanted to do these experiments were just published this past year, the individual cell filling experiments. But I think those are really great experiments to do in all cases. But I really wanted to get the baseline work. Pardon? Okay. Yes.
Two questions. So, in the ear, like in the eye, there is a crossing from one side of the brain to the other side of the brain. Are there any other organs where there is this kind of crossing between the two halves of the brain?
Well, the somatosensory system, of course, shows a crossing, and the midline structures are bilaterally represented in the cortex, and so the midline structures are getting input from both sides of the both sides of the brain. In most vertebrates, the eyes, the projection from the eyes is primarily contralateral, and there's in the vertebrates where there's convergence of the two eyes, you get an ipsilateral pathway. In the auditory system, the input is bilateral, but the representation tends to be contralateral. Okay, but the input is actually bilateral. Okay, so if you look at the output, for example, of nucleus laminaris, it goes to both sides of the brain. If you look at the output of MSO, it tends to stay ipsilateral, but then it combines at the level of the inferior colliculus.
And the second thing, in the retina there are two kinds of cells, analog cells and those that calculate. Is there something similar in the auditory system?
At the level of the cochlear nucleus, okay, most of the cells in any given area, at the level of the ventral cochlear nucleus, because it has to do this rapid timing, okay, it preserves the time. Its job is to preserve the timing of the input. And actually, the phase locking gets better in these cells that get 2 or 3 or 4 inputs from the same place in the cochlea. And we understand a little bit of why. But I would say that this is almost a digital system at the level of the ventral cochlear nucleus. Now, a lot more is going on in the dorsal cochlear nucleus, where there are many, many cell types, and they're clearly integrating a variety of kinds of both afferent information and downstream information from the colliculus and other places. But the system to be able to calculate microseconds differences in time of arrival of spikes, where those differences are less than the width of action potential, has to keep everything pretty pure.
I think we should...
One more?
One more.
He's been...
You suggested that the culture may be involved in these changes. So what I'm asking is, did you or anybody else in the field try to see whether there is sufficient amount of calcium left
Yeah, I think that's a great experiment, and I have been looking for a postdoc that wanted to take on those experiments for about 3 or 4 years. The techniques to do those experiments have only recently emerged in ways that are sufficiently satisfactory. We were talking today about some of those techniques, and all I can say, I'm looking for somebody that wants to do that. I'm hoping to find somebody that wants to do that. Yuan may go on and do that, but she's gotten really interested in Fragile X protein and its regulation of other proteins. And the question is if I'll find them or they'll find me before I close my lab. Frankly, that's the goal. I don't know. Hopefully other people will use this system to try and understand dendrites. Few people have. Most people are interested in cortex and spines, which is good for me. It leaves me alone. Thank you.